Saturday is
Friday after 9, the library closes. Actually, after 9 is probably best. I'd like to see in the room. Oh wait, you don't know. Perfect. Okay. So we'll just do 9 to 11. That's so funny.
Okay, let's get started. So, first of all, on Monday, I'm going to want the partners and what your hormone is. That's all I need. Just what the hormone and what aspect of the hormone you'd like to do. So really, what Monday is all about is making sure you have a partner and you're getting started. Okay, so those of you who have not found a partner yet, I'll try to remember to say this at the end of class as well. Maybe you can all meet up here in the corner and find the partner to work with. Okay? And you can either, on Monday, you can either give me just a piece of paper with your 2 names and the hormone that you're going to be choosing, and then hopefully a little bit deeper than that, but doesn't have to be that much deeper. And then also, or you can send me an email. Either way. Okay? Second thing is this Friday, the seminar is going to be given by a colleague of mine, Glenn Crossan. So this is the one that I highly recommend going to. This is the one lecture, Friday lecture of the year that's actually going to be about hormones, or at least partially about hormones. And he does a lot of really cool work on penguins and skuas and all kinds of crazy animals, crazy birds. So it should be really entertaining. But come early, because if all of you show up, there might not be enough seats for everyone. So if you want a seat, show up 5 minutes early. Okay, lastly, I have the exams, which you probably are all interested in. So just a little bit of information here. So my record is still intact. Nobody has gotten 100% on any of my exams yet. The high this time was a 57. The low was a 27, but more importantly, the median was a 47. So what does that mean? So my intention is that the median will be the cutoff between a B and a B plus. Okay, so that'll give you an idea of approximately where you are. I'm not going to curve this exam. What I will do is curve the final grades at the end of the class. Okay? So if you didn't do as well as you would like, do not panic. First of all, I am a firm believer in progress and improvement. I do recognize that most of you have never taken an exam like this before, and sometimes it takes looking at one to figure out how to answer things. So if I see substantial improvement between the first exam and the second exam, your grade will not be a straight average. I will weight it heavily towards the second exam. Okay? Second of all, remember that people generally do really well on the project, and the project is a way of a lot of people will actually improve their grade. And so I just want everybody to remember, too, that I'm not here to fail anybody. So that's, I want all of you guys to do well. So don't worry about, one of the things I do not want you to do is to take your score and divide it by the total to see what your average or what your percentage was and think that that's just your grade, because that's not the way I'm going to look at it. It's not the way the grade is going to be made. So look at it in terms of where you are relative to a 47. Okay? Lastly, the exams are graded by one of my graduate students and Michael. I have not looked at them. If you think that you have been unfairly graded, I'm happy to take a look and give a regrade. But just a caution, I will give a full regrade. So I'll read throughout the entire exam. So look at your each question and try to figure out whether or not you think that the entire exam has been graded fairly. So the reason I say this is that oftentimes when we're grading, we take a look at the first question and we're like, yeah, it's close, not quite exactly, but I'll give them the benefit of the doubt. And then you look at the second question, it's like, it's close, but I'll give them the benefit of the doubt. And you look at the third question, and you're like, no, okay, I gave them the benefit of the doubt twice, not this third time. But if all you do is look at the third question, you're going to say, oh gosh, I could have gotten another 2 points. But when I look at it, I might have the exact same feeling. The other thing to think about, I hate to say this, but this actually did happen. A number of years ago, a student asked for a regrade on one question and thought that, and I agreed with them, that the first, their answer was not what I had been looking for, and so that was not on the answer sheet that I gave the student, my grad student, a grade, but it was correct. So I gave her an extra 3 points for that question and went to retotal and found out she had lost 7 points. That the grad student had mistotaled the original exam score. And so, she came to me and said, how can I lose 7 points? And I said, well, you didn't earn those 10 points in the first place. So now I can't give them to you. So just look at the entirety of the exam before you ask for a regrade, okay? All right, so let me quickly go over. I don't want to spend too much time on this because I have a lot of really cool stuff to talk about today. But I do want to just sort of tell you about what kinds of things we're thinking about. And I know not all of you answered every one of these questions, but so the first question on the regulation of the hypothalamic and pituitary when there was no portal vasculature. I was looking for essentially 3 points. One was that you need to have an increase in the amount of releasing factors coming out of the hypothalamus, because they have to go all through the circulation. They can't just go straight through the pituitary, through the portal system. The second thing I was looking at is that the pars distalis is going to have some change in either the receptors, either the KD of the receptors or something, so that they can recognize and be more sensitive to a lower signal coming through the rest of the body. And the third thing I wanted you to recognize is that the pars nervosa, part of the pituitary, would not change, because that's all still neuronal. Okay? Question number 2, which was on looking at the impact of a new hormone on milk ejection. And really, the question had very little to do with milk ejection and oxytocin. The question was really about direct effects and indirect effects. So if this new peptide had a direct effect on milk ejection, it was looking at something about being an oxytocin mimic, or an agonist. If it had an indirect action, it needed something about how it increases the amount of oxytocin that's been released. If it was a permissive effect, something about increasing the number of oxytocin receptors, or some way in which the oxytocin was working better. And then the technique that you would use, we were looking for HPLC, because that's how you would have isolated a new hormone. Question 3. Looking at positive and negative control. I should just write this up on the board. So, what was it? So we had X, and I was looking for Y, which had positive control, and W, which had negative control. And then the negative feedback, in order to maintain it, you'd have a negative impact on Y, but a positive effect on W. Because we need to increase the amount of W in order to decrease the amount of X. Okay? If this was actually a positive effect, that was part B, then what you get is a runaway regulation of X, so that only Y would be controlling the amount of X. That's what I was looking for. And then for the last part, if you have Z, that is permissive on, permissive on Y, And what happens if you lost it? Now, you wouldn't have any effect of Y, so you'd never get X being released. You'd only have this negative function. Okay? The question 4, so you're looking at, this is the one with the mutant cell line that no longer responds to a peptide hormone. Really what I was looking for, again, is something about the understanding of what a tyrosine kinase receptor is and where the different domains are. So the part A was what happens if the receptor's KD is dramatically elevated. Well, an elevated KD means a decrease in affinity, which means there must have been a mutation in the hormone binding domain. If the receptors were not on the cell surface, but instead in the cytoplasm, that means that there must not have been movement of the growing mRNA to the endoplasmic reticulum, where it could be put into the membrane. And so therefore there must have been some mutation in the signal sequence. For part C, none of the receptors were on the cell surface, but instead were in the bath surrounding the cell. That means that as the receptor was being produced, it didn't get stuck in the membrane, it just went all the way through like a hormone would, so there must have been some mutation in the membrane spanning region. And then D, if you had extra amounts of steroid that are known to regulate the levels of this receptor, and then the peptide hormone returns to normal, that means that there's something, either there wasn't enough steroid in the first place to help increase the amount of this receptor, or there was some problem in the hormone receptor element, some mutation there where there weren't getting enough regulation. Okay, question 5 was about a young kid getting bitten by a spider in the rainforest that wasn't growing properly. So what effects if GH levels were normal? So the idea here was just to look at growth hormone coming down to IGF-1. And so if this is, so what was part A? If GH's levels were normal, and IGF levels are normal, but the child stops growing, so this is where growth is, then we're looking at is that IGF-1 is not affecting growth. So this is probably an IGF-1 antagonist, but it's not affecting the regulation up here. In part B, GH levels are normal, but IGF levels were elevated, and the child was growing a lot, so this levels are elevated, then that means that this must be a GH agonist. So it's increasing the amount of IGF-1 that's being produced. Part C, GH levels were elevated, but IGF levels were low. So we have high GH, low IGF, then that's really indicating this must be a GH receptor antagonist because the child is not growing. So this signal is not getting through because there's no IGF-1 being produced. And then finally, IGF levels are low and so both growth hormone and IGF levels were low. So the entire system is downregulated. So what we were looking at then was we have somatostatin. So it must be a somatostatin agonist that's shutting down the entire pathway. Okay. 6, the astronaut who ate some stupid plant. You never see this on science fiction movies, right? So we expect that the idea here in terms of what is trying to recognize that what you're looking for is something about vasopressin. And so if you recognize that the plant is probably mimicking vasopressin, then that's why you're getting these symptoms. And the other, because it must be an agonist for vasopressin, then other kinds of things you'd be looking for are other effects of vasopressin, like thirst and or increasing blood pressure. And so if you recognize that it was a vasopressin agonist, then the way to solve the problem would be to give a vasopressin antagonist, thereby inhibiting the effects of vasopressin. And if you changed it by saying that this was instead of an agonist, it's an antagonist, then the key to answering that part is to recognize that you're going to see the same symptoms both with very high levels of vasopressin and very low levels of vasopressin. But in this case, what you'd want to give is a vasopressin agonist rather than the antagonist to solve the problem. All right, so question 7 was, was all about, this is the rodents and birds saving energy by decreasing their gonad size. This was all melatonin. So trying to recognize, A, that melatonin is affected by day length, B, that the melatonin is inhibiting to GnRH, and therefore the role of N-acetyltransferase, if you have less of it, then what you're going to get is less of a break on the system. And so what you should see is then regrowth of the gonads earlier in the season. Or some people answered maybe they didn't even regress them fully in the fall, and that's also an acceptable answer. And so then if you have also 50% effectiveness of GnIH, you're going to get the same thing, because the GnIH is not inhibiting the system, so you should get regrowth of the gonads earlier in the year. And if you have the double mutant, you should just, those two things should work together, and you probably get even earlier regrowth of the gonads. And then finally, you're looking at the strain of rats that's hypothyroid, and really I was just looking to see if you understood the synthetic pathway of thyroid hormone. So if there was, if you could solve the problem by injecting hydrogen peroxide, then it must have been a problem with the TPO, the enzyme that produces a hydrogen peroxide. If colloid from an unaffected rat is working, that means there must have been a problem with thyroglobulin production, that it wasn't working properly. If you could solve the problem by adding clathrin, then it indicates there must have been a problem, since clathrin is there for reuptake, then there must have been a problem with reuptake after you formed the thyroid hormone. And if you could solve it by adding TRH, then it must be that there was a problem with TRH release from the hypothalamus. And finally, if inhibiting deiodinase type 3, if you remember, deiodinase type 3 is what prevents activation to T3, so there must be too much deiodinase 3 deactivating T4 in those animals. Okay, any questions? Yes?
For the colloid space, why is it related to thyroglobulin?
Because that's where the thyroglobulin is put. So if you take colloid out of the healthy rat and put it into this rat, you've taken healthy thyroglobulin and replaced the non-healthy thyroglobulin.
Yeah.
Yeah.
So more melatonin stimulation does not lead to the gonad size, and it's just early development.
Right, because you've lost the break. So if you have less melatonin, you have less break on the system. Okay, what we'll do is I'll hand out the exams at the end of class, so we don't take up any more time. All right. So this is where we were last week, or Monday. We're looking at the hormonal responses during pregnancy. So we talked about how we have this large increase in chorionic gonadotropin, which is the LH mimic, or HCG. And that is to continue driving the system so that the growing embryo and fetus still has a lot of estrogen and progesterone there. Another thing that gets produced, it's also really important, is chorionic somatomammotropin. And that's this top curve here. And that is, again, the somato meaning cell, mammary gland, tropin, stimulant. So what this is a hormone being produced by the chorion. So again, that's being produced by fetal tissue that is going into mom and stimulating both the growth of the mammary tissue and starting the production of milk. And so you can see it increasing over the course of pregnancy. So yet again, this is the fetus regulating mom's body. Okay? So then we get to the process of birth. So there's a lot of different parts of regulation of birth. But one of the first things that you recognize is that mom does not decide when birth takes place. The fetus does. And the fetus does this essentially through the glucocorticoid system. So there's two things. There's CRH that's coming from the fetus's hypothalamus, stimulates its pituitary to release ACTH, which goes to the adrenal, and you get the release of cortisol. And cortisol then goes to the placenta, where it stimulates the production of a couple of prostaglandins. The prostaglandins then help regulate this system here, but they also, importantly, stimulate the production of P450C17, one of the P450 enzymes, which is converting progesterone into estradiol. The progesterone that's coming out of the placenta normally is prohibiting and inhibiting contractions. And so when you start converting the progesterone into estradiol, you remove that brake to the system. The estradiol, also being produced by the placenta, travels into the uterus and starts stimulating contractions. At the same time, CRH is being released by the placenta that goes to mom's brain and starts stimulating her release of cortisol. Okay? So the trigger for this birth is being driven by the hormones of the placenta and regulated through the fetus's hypothalamic pituitary adrenal axis. Two other big aspects of this. One is that cortisol also is incredibly important for organ formation right in the last couple of weeks of birth. One of the biggest ones is in the lungs, and it stimulates the production of surfactant. Surfactant is a fluid that coats the inner lining of the lungs and allows and dramatically increases the transfer of oxygen from the air into the capillary tissues. So without surfactant, it's going to be real trouble. So this turns out to be one of the biggest issues that was solved in medicine for premature births. So most infants born prematurely died until fairly recently. And one of the big things they discovered is the reason why most of these preemies died was because they weren't able to get enough oxygen. They weren't breathing properly. So one of the first things they do when a preemie is now born is they inject cortisol. And the cortisol is there to stimulate the production of surfactant. A couple of years ago, I talked to a neonatologist who did a lot of this work, and he says, yes, they still have to inject the cortisol. Nobody wants to, because there's all other kinds of problems that high levels of cortisol will create in this baby. But they still don't have a way of stimulating the production of surfactant other than giving cortisol. So there's actually quite a bit of research still going on today on trying to figure out how can we get the production of surfactant without injecting cortisol. Okay, so one other new hormone that is really important during this process and is produced by the placenta, and that's a hormone called relaxin. And that goes to the uterus and helps relax and the opening of the cervix and allows the birth to take place. Okay? Yeah.
So, at what point does the release of cortisol from the fetus result in the conversion of progesterone into estradiol?
That's a terrible picture, isn't it? I'm sorry, when does this,
Because you mentioned
the
how
progesterone into estradiol?
Yeah.
So this process is taking place because the cortisol is stimulating the prostaglandins. So it's really when the fetus decides it's time to be born, that's what's stimulating the prostaglandin, which stimulates the production of this enzyme that gets the conversion.
But then we talked about how cortisol is also used for organ
Yep.
maturation. So why doesn't that convert progesterone into estradiol? Like, isn't the fetus continuously releasing cortisol? Or?
Yeah, but it's building up right close to birth.
Oh,
So it's only right in the last week or two that you start getting this increase.
Oh, okay.
Okay? Yeah. Yep.
I missed it. What does CRH cause the mother's brain to produce?
More cortisol
Oh, just
for
more
her.
cortisol.
Yep. Yeah.
Can really high levels of stress cause a mother to go into labor?
Yes. And it's not just humans. So one of the really cool, I don't think I told you guys about the frogs and the snakes, have I? Yeah, I did. So yeah, the snake will go up and the stem and the eggs of the frog will detect those vibrations, and the frog embryos will start to release a huge amount of CRH, which increases not cortisol but corticosterone in them, and they will immediately hatch. Okay, and it's true even in humans. You can get premature labor because of stress. So you can see this sometimes in late term women who get into a car accident, for example. Very shortly after the car accident, all of a sudden they'll start going into labor. And it's probably done by the cortisol. But it's all primed because the baby actually decides when this happens, not mom. Yeah.
So the cortisol in the mother's brain, is that what's responsible for maturing the fetal organs, or is it do something else?
No, it's the fetal cortisol. So mom's cortisol is actually helping prepare the uterus, etc., for getting ready for birth. Yep.
And so the CRH and the relaxin are released from placental cells in the fetus that are going to affect mom as well?
Yes. So people know how the placenta is created. So you got the uterus, and the uterus has all of these what are they called? But the tissue has pits and things like this. And so then the egg, which is not because by the time it gets to the uterus, it's generally at about the trophoblast stage, so probably 100 to 200 cells. And it implants inside the uterus and starts to even grow, and at that point starts to go through gastrulation. And when it goes through gastrulation, you start to produce the different cell layers. And one of the first things that gets produced are these what are called the extra embryonic membranes, and one of which is the chorion. And the chorion builds in, and as it starts to grow, what you see is the embryonic tissue coming in and meshing with mom's uterus tissue. And so that's the chorion that's inside the placenta, and that's what's producing all of these hormones. So the placenta itself is a mixture of mom's tissue and fetal tissue, but mostly fetal tissue. Yeah.
So is the cortisol stimulating progesterone conversion directly from the fetus, or is that stimulated by the fetus but still produced by the mom?
Both. Okay? So the fetus is both producing its own cortisol and stimulating mom's cortisol. Yep.
Is because the placenta is half mom's but mostly fetal, is that why the mother doesn't usually attack the fetus? Because it still thinks it's kind of their own tissue?
That's part of it. The other thing is, mom sometimes does attack the fetus.
Yeah.
So there is I can't remember the name of the syndrome, but there can be. And part of the reason is that the tissues are so close that what we're discovering is that a lot of the cells from the placenta from the fetal part of the placenta will break off and start circulating all through mom's body. And then that seems to be one of the hypotheses for why that happens, is that it helps train mom's immune system to not attack it. But you can actually take blood from mom and find fetal cells circulating. Remember, parasite. Yeah.
So is the progesterone acting as a constant break from contraction?
Yeah.
So to have contractions, you both need the decrease in progesterone and the increase in estradiol?
Correct. So you need to get rid of the brake
Yeah.
and also add the gas.
So it's tonic.
Well, yes, a tonic inhibition, but
Okay.
so the progesterone is continually being made. Yep. Yeah.
So if you just got rid of progesterone but didn't have the estradiol, what would happen with contractions?
Well, you wouldn't start driving contractions because you wouldn't have the gas, and it would be a disaster because you also wouldn't be producing relaxin yet, and so you couldn't actually push the baby out. Yeah.
Can you explain one more time how CRH and relaxin stimulate body?
So these are both being, their production is regulated, being produced by the placenta, and as of right now, we don't know what that signal is to stimulate this production.
Oh, okay. So that's
Okay.
from the mother.
No, this is from the fetus, but from the placental part of the fetus.
Oh, so the fetus is releasing CRH and relaxin.
Correct.
Which goes to the mother.
Correct.
Oh, okay.
Okay? Yeah.
Does this have to do with why twins are often born early? Like, do they produce more cortisol together?
Oh, that's an interesting question. I don't know the answer to that. My understanding has always been that the reason twins are often premature, it has more to do with sort of space and growth inside the womb. But it wouldn't be surprising if you have two placentas, both producing relaxin and CRH, that you just all of a sudden start to stimulate it early. But I don't know if that's ever been looked at. Yeah.
Is P450 only produced when it's time for birth?
Yeah, so these prostaglandins are stimulating the production of that enzyme. Yes.
Sorry, this is kind of a silly question, because I understand there are other factors contributing to like the actual process of contraction of birth, but if cortisol is acting as like that intermediary between contractions and birth itself, and also leads to fetal organ development, and so that can be produced, what are the scenarios where it's not a matter of cortisol being produced and you have premature babies that don't have those factors?
So most of the premature babies are not actually born. So that's the thing to remember about preemies. They're usually delivered by C-section.
Oh, because like an ectopic pregnancy.
Things like that, right? So especially the really young preemies are generally delivered by C-section, not normal birth. And so they haven't gone through this process yet. And that's why they don't have surfactant. Okay? Okay. Once the baby is born, we have to feed it. And here we are, the mammals, right? So we have mammary tissue. So I'm going to come back to the mammary tissue. So here's a really more detailed picture of the mammary tissue than we had earlier, where you have these these milk ducts and all these alveolar sacs that are producing the milk. And so this is part of what we showed last time, where you have these myoepithelial cells. But here it is in higher detail, where you have, these are the epithelial secretory cells. These are being regulated by prolactin. And these are the contractile cells, the myoepithelial cell bodies, and these are being regulated by oxytocin to stimulate the production of milk. And if we look at this in even more detail about how that's actually regulated, we get this figure. Okay? So, really complicated figure, but I love it because it really shows what's going on. And as soon as you understand what's going on here, it'll start to make a lot of sense. So, first of all, we have progesterone coming from the corpus luteum. It is inhibiting the mom's prolactin inhibiting factor. So you inhibit the inhibitor, what do you get? An increase in prolactin. And so the prolactin is going to be released from the pituitary, go to the breast tissue, and stimulate the increase in breast production, along with the somatomammatropin, and also starting the production of milk. At the same time, we can get estradiol also going to the pituitary and directly stimulating prolactin synthesis. So we get both of these things. We get a direct effect at the pituitary, and we inhibit the inhibitory factor to get more prolactin release. Okay, so we've got milk. Milk is ready. Baby comes up, attaches to the nipple, and starts to suck. That sucking stimulus is detected by the nerve cells in the nipple, which sends the signal through the brainstem, up through the spinal cord, directly up into the brain. And one of the things that it does, it goes directly to the hypothalamus and stimulates the release of prolactin releasing factors, as well as inhibiting the prolactin inhibiting factors. So we're going to get an increase in the amount of prolactin that's going to be produced. At the same time, this signal goes directly to the other part of the hypothalamus that sends the oxytocin cells, and the oxytocin is going to be released from the pars nervosa. And this is going to take place in the order of seconds. And so the oxytocin is released in the bloodstream and goes to the breast. You're going to get the contraction of those myoepithelial cells, and you'll get the release of milk into the baby's mouth. So then the second process starts where you get the increase in the prolactin, and that will then stimulate the replacement of the milk. And that generally takes about 3 to 4 hours to resynthesize milk. Yes?
So in this case, prolactin is sort of like the long-term effect, whereas oxytocin is the one that produces.
Yep. So the oxytocin is going to activate all of this. The prolactin is going to be building it and replacing the milk stores. Now, at the same time, it doesn't actually have to be necessarily a sucking stimulus, but other things can activate these oxytocin neurons as well, including a visual stimuli and even auditory. So, nursing moms often have to have pads that they place over their nipples while they're breastfeeding, because walking into a grocery store and seeing a baby crying or seeing a baby nursing will immediately activate those oxytocin neurons, and they will start to eject milk, and it will start to leak from their breasts. Even just hearing, you don't even need to see the child, but just hearing a baby crying will immediately stimulate these neurons, and you will get the release of oxytocin, and they will start to eject milk. Yeah?
Maybe I missed this, but what is signaling the increase in the prolactin releasing factor and the increase in the inhibitor? Like, what causes that?
This is a neural stimulus directly from the brainstem.
Okay. And is that caused by like environmental cues?
This is these environmental, yes, from the baby suckling. It's this suckling stimulus that sends that neural signal right up to the brain.
But isn't there milk there before the baby starts nursing?
Yes. So that's this pathway to actually produce milk. So we've got two pathways here, and that's part of the interesting part here. We've got two pathways: the pathway to produce milk and the pathway to release milk. And so the pathway to produce milk is by prolactin. The pathway to release milk is the oxytocin. Yeah?
Is there negative feedback in this?
No, because the thing that will stop this whole system is the loss of this suckling stimulus. So that's one of the things that happens with humans a lot, is that, especially in the modern world, is that women don't breastfeed often enough, because basically you need to be breastfeeding about every 3 to 4 hours. And if you don't do that, you're not getting sufficient stimulus, and so the whole system starts to ramp down. And so that's one reason why a lot of professional women, for example, when they go on trips and things like that, they will pump milk in order to continue that stimulus, so that they're continually producing that milk, so that when they return, they can still breastfeed. Otherwise, if they stop doing that, because baby's not there, they lose those stimuli, and when they lose those stimuli, these pathways start to become less sensitive. Yeah.
I know that most times you can get away with not having any kind of like birth control because of actually breastfeeding. Is that because of progesterone levels?
Absolutely.
And that's why they can't actually release any eggs and go through the normal cycling that we went through last class?
Yep.
So if you decrease the amount of breastfeeding, that's when you start ovulating again.
Correct. So that's one of the big things, is progesterone is not only, remember, stimulating this, it's also going to the hypothalamus and shutting down GnRH release. So, you know, I forgot to talk about this last on Monday when we started talking about why do women have menses. And one of the interesting things that's come out recently is, how many menses do you think the average woman went through during her lifetime, pre-modern? You want to take a guess?
Is it significantly fewer due to
So why would it be fewer?
stress?
Probably not, yeah?
Because basically women were pregnant whenever they could get pregnant.
Yeah, you get pregnant whenever you can, and then how long, do people know how long? So pre-modern, we base most of this on pre-modern societies today, but how long do the women generally breastfeed? No.
Like 5?
4 to 5. So for 4 to 5 years, you're breastfeeding, and thereby you're shutting down GnRH production. And since most of those women were probably almost always pregnant, modern estimates are that probably the way our evolution designed us is that probably women were only going through maybe half a dozen, at most maybe a dozen menstrual periods in their lifetime. So it changes our perception then about this lack of the blood and lack of iron, right? I told you, remember, that there was a number of pieces of information that maybe that flushing the system and getting rid of the pathogens may not be. That's part of the evidence that people suggest. To say, look, yeah, that's great, and this may be very important for modern women that don't breastfeed for 4 to 5 years. But if you're continually suckling a baby for every 4 to 5 years, and then you get pregnant again very shortly thereafter, this is not something that actually evolution would have worked on very, very often. Okay? Yes?
I understand how if you stop suckling, you stop contraction, but how exactly is milk production shut down?
You just stop. You stop producing some of the progesterone. Stop inhibiting the prolactin inhibiting factor, and so your negative break starts to increase.
Okay.
Yeah. So there's other reasons why you want to suckle a baby, and so at this point, the recommendations from all OBGYNs is that all women should attempt to, unless there's another reason, should attempt to breastfeed for at least 6 weeks after birth. That didn't used to be the case, and it turned out to have rather tragic consequences. So back in the 40s and 50s, doctors decided that breastfeeding was actually probably not sanitary. And so they recommended that women not breastfeed at all. And it probably had nothing to do with the fact that companies were starting to produce formula and wanted to sell it. But anyhow, the recommendation was that you not breastfeed, that you go immediately to formula. And it turns out that there's another thing that breast milk has, especially at the beginning, in the first couple of days. It's very high in protein and low in fat. And it's called colostrum in the first couple of days. And it turns out to have a huge number of antibodies. And one of the important antibodies that turned out to be transferred from mom to the infant were the antibodies against polio. And so back in the 50s, as the doctors were trying to recommend, okay, it's not sanitary, you shouldn't be breastfeeding, we all of a sudden had that massive increase in polio, a huge polio epidemic in the country. And so that's one of the reasons why they recommend, unless you have other medical reasons not to, why they want all women to try to breastfeed for 6 weeks, to provide some of those other things to the baby. Which brings to me a very just interesting fact, which is what is the milk content or fat content of milk? So I found this wonderful website. Let me see if I'll increase this a little bit. So here is just the content of fats in different kinds of milks. And you can see here's human. Human milk has about 40 milligrams per milliliter of fat. And it's actually, human milk is quite low. But it's still higher than cow milk. So here's cow. This is what most of us drank as kids, and maybe a lot of us drink now. That's at 34. But some of our other animals, like goats, they're up around 48. So they actually have a higher fat content than cow milk, which might explain why goat cheese tastes so differently than cow cheese, because there's very different fat contents. But then you have some really crazy things, like dogs are just our pet dogs have more like 93. So they have over twice the amount of milk fat in their milk than humans do. And then you get, I don't know if you're familiar with the Sami in Lapland, up in Finland, but their diet is primarily from milk from reindeer. And so they herd reindeer. And it turns out the reindeer have 171 milligrams. And then you look at some of these crazy ones like harp seals and blue whales. So these whales that are, their babies are putting on sometimes as much as 50 or 60 pounds of weight per day. And you look at they're 381 milligrams of milk fat in a milliliter of milk. Yeah.
How do whales nurse their babies if they're swimming?
Same way. They just sort of slowly swim, and the baby comes up and attaches to the nipple.
The babies can swim as soon as they're born.
Yeah. There's, like horses that can walk almost as soon as they're born. The whales can't. I'm told that they can swim, and that's pretty easy. But what they don't always know is how to breathe. And so apparently, mother dolphins, one of the first things they do is have to help the baby get up to the surface and breathe. And after a couple minutes, the baby quickly learns how to do this. But that apparently is not instinctual. Yeah.
What's the significance of the ash column?
The ash, that's the amount of, I think that's protein that after they essentially burn it. So I think that's protein content. Yeah.
At least how I've seen ash before in like different nutrition classes, it's usually like extra minerals that they
okay.
can't measure that's just left over after everything's burned away.
Yeah. That's probably a better answer. Okay? All right. So this is a good transition, because the next lecture and a half, now we're running out of time, we're going to talk about some of my favorite things in the class, which is I've mostly talked so far about mammalian reproduction. So for the next little while, we're going to start talking about some mammalian reproduction, but really cool mammals, and do things entirely differently than humans do. and some non-mammals. And one of the ways we're going to start is just thinking about how different species regulate breeding. And there turns out to be two major kinds of breeding. There are what are called the associated breeders and the dissociated breeders. And so here's a graph. This is when mating occurs. If mating occurs at all times of the year, then there's generally very high and constant levels of gonadal activity. But for some, if you look at testosterone, for associated breeders, you get a big peak of testosterone right at the time of mating, which then decreases. But then a dissociated breeder, you will have very low levels of testosterone during mating, but an increase after, not associated with mating. And so if you look at this, this is the time during the year. And so you have mating occurring here. Spermiation, or the production of the initial aspects of sperm production, would take place right near mating. And spermatogenesis, the actual production of mature sperm, is going to take place right before mating. This is the associated pattern. And you can see the plasma androgens are going to peak right at mating and then decrease for most of the rest of the year. The dissociated pattern, instead, mating appears here, but spermiation and spermatogenesis take place at a completely different time. And so the plasma androgens are up high here, not during mating. So the question is, why would you have these two different types of systems? And the answer seems to be that you want an associated system when you want the sperm production at the same time as mating occurs, which is for most mammals. But you want them separated if mating occurs at a different time of the year than you want your sperm production. So for example, a hibernator. A hibernator might pair prior to the winter. So they're mating here, but they're actually producing their sperm in the spring, and that's when you want your high testosterone. So the key aspect of this is that you want your high levels of testosterone when the sperm is being produced, not necessarily when mating occurs. Okay, so we talked about induced ovulators like the cats. The same thing is true of the females. Similar thing is true of the males. The males have to have their sperm ready to go when they see a female, and so they are actually dissociated breeders because they are producing their sperm and having their high testosterone levels different from when they actually mate. Okay? All right. The other big aspect of things that testosterone does is we have to worry about the production of how do you, the actual regulation of the gonad and the gonad development. And there are three major types of gonad development. So there is the genotypic sex determination, where you actually have an X and a Y and genes on there. And we're going to talk about that in a minute. Another one, though, that takes place in a lot of the reptiles, especially the turtles, the alligators, and a lot of the lizards, is temperature-dependent sex determination, in which you have the egg and the sperm joining to make the embryo, and whether or not you make an ovary or a testis will depend upon the temperature of incubation. So here's an example of what it looks like, and it's very different with different species. So this is a lot of the alligators and a bunch of the lizards. And if they're incubated at approximately 28 degrees Celsius, then you're going to get 50-50 ratio. So 50-50 ratio of males and females. Whereas this is the proportion of males. So this is all females, and this is all males. So if you incubate these eggs at a high temperature, you're only going to get males. And if you incubate them at a low temperature, you only get females. Now, in the wild, this doesn't particularly make any difference. So anybody who knows anything about alligators, what they do, or a lot of these turtles, they'll go in and they'll dig a hole in the soil and then lay a whole bunch of eggs. And those eggs are all incubating, and they're producing their own temperature. But the eggs in the center are not only getting their own heat, but also the heat from their neighbors. And so they end up being heated higher than the eggs on the outside. So the outside of the egg mass is going to mostly produce females, and the inside of the egg mass will mostly produce males. And so what you generally get is the 50-50 ratio overall of males and females being hatched. So why should we care about this? Well, I should point out that other species, like a lot of other turtles, are also have the same kind of curve, but are the opposite. So low temperatures makes males, high temperature makes females. Some of them don't care, they just produce a 50-50 ratio no matter what. And others have a peak right here at about 24 degrees, in which case they produce all males at the medium temperature and all females at both the high and the low temperature. So a lot of this temperature-dependent regulation. So one of the ways in which this was discovered was very interesting. So a lot of the sea turtles are highly endangered. And at one point, and one of the reasons they're endangered is because they haul themselves up on a beach, they dig a hole, they lay their eggs, and they go back out to sea. And then they leave the eggs there to hatch. And a lot of people would come by, dig them up, and take them back for turtle egg soup. And so a lot of these sea turtles, populations were being decimated. So one of the ways in which conservation biologists were trying to solve this problem is that they would watch these females come up on the beach, lay their eggs, and then they would go out as soon as she went back to sea, dig up the eggs, and take them back into the lab and incubate them. And as they were doing this, and then they'd incubate them, they'd hatch, and they'd take the hatchlings out and put them back in the ocean when they'd hatch. And the populations continued to crash. They kept losing, they have fewer and fewer and fewer turtles coming back to breed, and people couldn't figure out why. And it turns out that that species of sea turtle had this type of temperature-dependent sex determination, and they had thought, well, if we incubate them at a little higher temperature, they'll develop faster, and we can get them back out in the ocean faster. And so they were incubating all of those eggs at a higher temperature, around 30 degrees C, and they were producing exclusively males. And so they did nothing for about 15, 20 years. They did nothing but put males back out into the ocean. So this is, by the way, something that has a lot of conservation biologists really, really worried. Because one of the other things that we can see is that what would happen if your soil was actually 1 or 2 degrees warmer? And that's what seems to be happening with global climate change. And so people are really worried that we're going to lose species because we're going to be basically producing almost exclusively males in the wild. Yeah?
Do we know why for some species colder temperatures produce females and higher produce males?
We do not.
So it seems arbitrary.
Yes, it does. And as far as I know, there's no, you have to actually go in for each species and figure out what it is. Yeah.
Is there a benefit to having this type of temperature regulated sex determination rather than just genetic sex determination?
I don't know. Notice that these are very ancient taxa. Alligators, crocodiles, turtles, they, are quite far away from us, from mammals. In fact, birds are closer to us than these species in terms of the evolutionary tree. And so it may be, and I haven't seen anything written about this, but it may be that the actual gene determination of sex came later, and that this may be more ancestral. So it's clearly worked. These species still exist and existed well before mammals arose. So this is a very effective system. The question is whether it can continue to be effective when we heat up our planet. Okay, so that is temperature dependent. How are we doing? Oh, I still got time. Okay. So the next one I want to talk about is behavioral sex determination. In this case, what generally happens is that the egg and the sperm fuse, creating the embryo, and generally, in most species, it can be reversed sometimes, but all embryos become female, and they start producing ovaries. And then at some point during the lifespan, the ovaries transition and become testes. And so here's an example. This is the blue-headed wrasse. This is the female, and this is the male. And what you have in this, a lot of the species that do this are actually reef fish, but you have others, a lot of amphibians can do this, as well as other kinds of fish. And so all of the embryo is going to produce females. And this is, a lot of these are harem species. So you have one male that has a half a dozen females that he guards and fertilizes their eggs. But they're much more colorful, they're often bigger, they're involved in trying to maintain this harem. They're also, because they're more colorful and they're bigger, they often are a better target for predators. And so if this fish gets knocked out, what generally happens is the largest female in that harem starts to produce a hormone called 11-keto-testosterone. And the 11-keto-testosterone all of a sudden starts to flood the brain, and it goes to the gonads and starts changing the ovary into a testis. And then once the testis is created, then the testis starts to produce testosterone, and you start to get the production of the different coloration in the different muscles. So we're actually converting the female into a male. Now, is that cool or what? Yeah.
Now, does something similar happen? Because I know some reef fish they change from male to female.
That also can happen. It's far less common.
Yeah.
Yep. Yes?
How would only the largest female be producing this?
Great question. Nobody knows yet what the social signals are to tell one of the females to change and the others not. Yeah?
Could you explain how the female senses the loss of the...
Nobody knows.
Are they sort of paired at least?
No, because it's a harem species. So all of a sudden the male's not around. People have done research to figure out how long the male has to not be around before the change starts. And if she starts to change and then the male comes back, I think in most species she will continue all the way and become a male, even though now there's a male around. So this is starting to get really old now. But I think probably most of you have seen this movie. What does this movie does this remind you of? Jurassic Park. So in Jurassic Park, written by Michael Crichton. Thank you. Michael Crichton was a physician, but mostly a biologist, and he knew about this. And some of the frogs will do it. And so if you go back and you ever look at that movie, they talk about the fact that they only had certain sections of the dinosaur DNA, and there were parts that were missing. And so they used frog DNA to put in to complete the missing sequences. And those sequences, what Crichton said, or postulated, contained the mechanisms for converting. And so if you and then they're so proud of the fact that they don't have any problem to do this because they've only produced female dinosaurs. And then they do want to produce a female so they can impregnate them and get more dinosaurs, but they can regulate when the breeding takes place because they don't have any males, except for the fact that a bunch of them convert into males. That's the whole premise of a bunch of Jurassic Park. And he knew this, and he did that intentionally. Think about what humans would be like if we did this. It's pretty impressive. I sometimes say that if I had to choose my career all over again, I'd be studying these fish and the sex changing aspect of this. It's just to me, it's just really, really cool. And if you think it's cool, we have already gotten commitment. So the Rotor Lecture, which is a lecture in the spring hosted by the biology department. The speaker in the spring is going to be Professor Russ Fernald from Stanford, who is one of the pioneers of looking at this sex change and sex changing fish and how the endocrinology works. So I highly recommend looking at that. It's sometime in April. Okay? All right. It gets even cooler than that. Okay? So these are just still 2 species. There's a lot of species, especially fish, that have 3 sexes. So here, this ugly little critter is called a plain fin midshipman. And it has 3 sexes. So it has females, and then it has males that look like this. They're about 1/3 to 1/2 again bigger than females, and they develop this sexual organ. So it's called basically a singing. They call it a singing organ, but they really hum. They really don't sing as we think about it, but they do make this big humming sound. And they do this to attract females, because what the males are doing is they are building. These are fish that live in water that's only probably about this deep, with lots of rocks and things on a sandy soil. And so what they'll do is they'll go underneath the rocks and dig out and excavate a little cave underneath these rocks that are probably about this size. And so they dig that little hollow, and then they attract a female. And the female will come in and she will lay her eggs inside that little cave. She'll leave, the male will come in, put his sperm on top of the eggs and fertilize the eggs, and then he will then sit there and both guard the eggs and every so often take water and squirt water over the eggs to continue to oxygenate them and allow them to grow. And so these are the males that are called macho males. Okay? Who says biologists don't have a sense of humor? Because there's also these males that are called sneaker males. The sneaker males look just like females. They're the same size, they do not have this humming organ, and they come up and pretend to be females. And so you can just imagine this macho male sitting there humming away, and 2 females come up. He just hit the jackpot, right? So the 2 females go in, except that one was a female and the other was a sneaker male. So the female deposits her eggs. The sneaker male pretends to be a female, goes in to lay eggs, but instead puts his sperm on top of the eggs and fertilizes the eggs, and then swims off. And then the macho male finally comes in and tries to fertilize the eggs that are left. And so you have 3 completely different sexes in this fish. And what generally happens is that your sneaker males are your younger males. And then over time, as they get older, they transition to become macho males. And they do that through 11 keto-testosterone. Yeah?
So isn't the macho male just kind of stuck taking care of the eggs because it just assumes that it's its own?
You got it.
Wow, it's just in real life.
Yeah. It's not just like, it is. Yes?
Aren't there species of birds that have sneaker males as well?
Not so much. Not in the same way.
Yeah, they don't change to be macho males, but there's like a subpopulation that kind of look like females sometimes and then have a tendency to do
Yeah.
the same thing.
We're going to talk about some of that. There's a lot of mammals that do this too. Okay? So, yeah. No, so one of the famous examples of this is slightly different. I was going to get to this in a minute, but this orangutans. People familiar with orangutans, right? What does a male orangutan look like? Huge, big, phalanges around its face, right? Well, the subadult males look like females. They do have functional gonads. They can impregnate females if given the chance. And they figure this out oftentimes in zoos. So what they'll do is they'll have the adult male in one exhibit and all the females and young adult males in another exhibit. And every so often one of those females will become pregnant, even though the males there don't look like these big males. And so there's been a lot of interest in zoos in trying to figure out how because if you lose the adult male, one of the subadult males will immediately start growing all of the secondary sexual characteristics of an adult orangutan. So what is preventing those young adults from growing all of these secondary sexual signals? And there comes a process that people call social castration. And people are still trying to figure out how that works, but it looks like actually the calls, the long distance calls of these male orangutans somehow breaks the system and the males and acts as an inhibition for the testosterone, the hormonal release in the subadult males. Okay, so back to the fish. You have questions?
Oh no, I was saying that there's a clip in Planet Earth or something about cuttlefish that were smaller than the dominant male that sort of act as females to then mate with the female cuttlefish under the watch of the dominant male. So they're not really different genetically, but they do use that tactic.
Yeah, the sneaker tactic is actually fairly common, especially among fish. So you get essentially 3 different sexes, and yet they are interchanged. And some of the sneaker males will never. And it seems to be a little bit of a genetic component as well, on whether or not. So a sneaker male is more likely to produce offspring that are sneaker males, and a macho male is more likely to produce offspring that are macho males. But there is a bunch of interconversion as well. Yeah?
Yeah, so I was going to ask if that appears to be a sex-linked trait or epigenetic.
Nobody knows yet, as far as I know. But come to the lecture in April, and we might have a better idea. Yeah?
Sorry, just to clarify, so if the macho males fertilize the eggs, would sneaker males come out because they're still young?
No, they can fertilize the eggs too. They have sperm. They just don't look like macho males.
No, I mean like the babies that come out, are they macho males or sneaker males?
A mixture, but if the father was a sneaker male, they're more likely to be sneaker males.
Oh.
And if the father's a macho male, they're more likely to be macho males. But it's still a mix.
And then eventually the sneaker males, once they get older, they convert back.
Some of them. So some of them will actually convert from a sneaker male to a macho male. Yeah.
And so why are they classified as different sexes if they're both males and they can both incarnate?
I'm going to ask you that question, but we don't have enough time to get into that. So I'm going to ask you that on Monday. Okay? All right. We don't have enough time. So before starting a brand new thing, we've talked about the temperature-dependent, the behavioral-dependent sex determination. On Monday, we'll start talking about the genotypic sex determination. Okay, so exams. What I'll do is I'll put A through, let's say, C over here. D through, let's say, L here. And then M through the rest of the alphabet we'll put over here. And those of you who do not have partners, please, why don't you probably meet over here in the corner, and you could find another person who doesn't have a partner yet.
I thought you were all the way over.
Your final score is on the inside of the top page.
Like 60% on the first thing. On the first thing, 55% on the second. Do you like just get it? Sure. Oh, sorry. Wait, what is it? D through D through L? So you send me an email about it? I don't think so. My partner might. I do not.
Okay. Unless there's 2 people thinking about a children's book, then yes, I think I got this. So I think it's a great idea.
Okay, great. Neither of us really do we want to
Absolutely, sure.
But she
Yeah, so you recommend dividing it out. Okay,
Something like,
how would I figure out kind of like what I gotta do?
I know, here, and also it's like they're all
transition to 47. If you're up there.